Hello and happy Monday morning. I'll be just getting on a plane to go to Chicago, and I hope that everybody had a good weekend and that you're ready to start talking about muscle contraction. So my goal in this lecture is to complete the muscle contraction, and then we'll talk about the energy metabolism on Wednesday. As you know, on Friday we stopped on Friday where we're talking about the neural junction, and we talked about how the action potential comes down here. It's going across the membrane of the axon, and it reaches the motor end plate where it's going to reach the axon ending. But we have this synaptic cleft right here, and we have the sarcolemma, these invaginations. And the action potential, we need a way to transfer it. What happens here, and this is the next slide to kind of show you different views of this. This is an actual myoneural junction, and this is a cross section. We've used these slides before. The one thing that we know is that these synaptic vesicles, which are right here, their purpose is to contain acetylcholine. Acetylcholine is a neurotransmitter. And what happens in this area is we have the synaptic vesicles that contain acetylcholine. Here it says containing ACH stands for acetylcholine. And we look at the bottom of the axon ending. What I like about this slide is it shows it as like a sieve. So we see here that the synaptic vesicles, they can move down here, and there's an area here where they have the ability to release acetylcholine, and it can drop through the axon ending membrane into the synaptic cleft. Let's look at that in a little bit more detail. Let's first look at this slide, and you can see an action potential coming down. Of course, this is my favorite slides, the Seeley slides, right? And as the action potential comes down, and this is not in your textbook, so you need to make sure that you make note of this. Notice that on the outside here of the axon ending, right above the motor end plate, there's free calcium. And that concentration of calcium is higher than what the calcium levels are inside the axon ending. So again, we have that concentration gradient where those calcium ions, they want in, but they can't get in. Here comes an action potential, and the action potential, notice that it opens up this calcium channel that's on the membrane of the axon ending, because the action potential is coming down here, right? It opens this up, and calcium is going to be able to flood into the sarcoplasm or cytoplasm of the axon ending. That calcium, that increase in calcium concentration, stimulates these synaptic vesicles, which contain acetylcholine, and acetylcholine, ACH, is an orange and a lighter yellow ball. That's acetylcholine. Notice it's contained here, but the calcium comes down. It stimulates the synaptic vesicles to move down to the axon ending and release acetylcholine into the synaptic cleft. Acetylcholine in the synaptic cleft then is preferentially binds to this receptor on the sarcolemma. The blue line here is the sarcolemma of the muscle fiber. Notice how acetylcholine, which is a neurotransmitter, because it's going to help transmit that signal, binds to this receptor opens it up, and that's a sodium receptor. And sodium, just like with the membrane here, high sodium on the outside, low sodium inside, we have the same conditions around the sarcolemma. So we open up the sodium channel, sodium, which is in the pink little dots, comes rushing in to reverse the charge along the sarcolemma and transfer the action potential, which is actually pretty amazing. So I told you we're going to talk about the steps of contraction and the steps of relaxation. Now, these steps are a little bit intertwined because the steps that start contraction also start relaxation. And that's what this portion of the slide is for. This shows a little bit a better view, a little bit zoomed in view. Here's the synaptic vesicle moving down, releasing acetylcholine binding to the sodium channel, sodium fluxing in. And this shows this is the synaptic cleft. That transfers the action potential. Because muscle contraction is a cyclic on-off, the steps that initiate contraction also stimulate relaxation. And in this case, here's acetylcholine on the receptor, but out here in the synaptic vesicle, or synaptic cleft, the synaptic cleft, there's acetylcholinesterase, or cholinesterase. Your book calls it cholinesterase. They're the same thing, two words for the same thing. And this is an enzyme. This enzyme breaks acetylcholine into acetate and choline. They can then be transferred by a pump here across the axon ending membrane to go back up into the cytoplasm. So a cholinesterase right here is responsible for stopping the action potential and closing the sodium pump because it plucks acetylcholine off of that receptor and then it's going to close. Acetylcholine then, because it's bound to the enzyme, then if we just release acetylcholine, it's not going to be able to go back up. It can't transfer across the membrane. So by breaking it into acetate and choline, it gets taken back up. Here's the choline, here's the acetate, and they're put back together into acetylcholine and then transferred back into the synaptic vesicle. So this process with an action potential coming down, let's just review this, causes calcium to flux in across the axon ending into the inside, into the cytoplasm of the axon ending. That stimulates the synaptic vesicles to release acetylcholine into the synaptic cleft. Acetylcholine binds to the receptor on the sodium channel of the sarcolemma. This is the sarcolemma, this is the muscle fiber down here. That opens the sodium channel and sodium fluxes in, and we have that reversal of charge like we did with the action potential. Acetylcholinesterase, or cholinesterase, pulls acetylcholine off of the receptor to close it back and breaks acetylcholine into acetate and choline. Those get taken back up because we have this channel here that will take choline and acetate back up, and they can go across the membrane and then they can be recombined into acetylcholine, and they are stored back in the synaptic vesicle, ready for the next the next contraction or action potential. Well, that's great. That's how we get the action potential to get transferred to the sarcolemma. And remember that the sarcolemma is sitting there, and as this action potential, it's going to go down and around the sarcolemma. When it reaches a T tubule, it goes down into the T tubule. The T tubule sets at the AI band juncture of the myofibril. Remember how on the myofibril we had the sarcoplasmic reticulum surrounding a myofibril? And sitting right next to a T tubule, because remember the T tubule is part of a triad. So a triad is one T tubule and two terminal cisternae. This is showing the membrane of the terminal cisternae or sarcoplasmic reticulum. That's what SR stands for. Here's the cytosol or sarcoplasm, and this shows a receptor that's plugged up. One of the things that we find in the sarcoplasmic reticulum, we talked about this in ultrastructure, if you remember, that there's a very high calcium concentration, 1,000 times more calcium in the sarcoplasmic reticulum, especially the terminal cisternae, than there is in the sarcoplasm. So again, another concentration gradient. Isn't that interesting? This concentration gradient, calcium cannot move across the membrane, and so we have to have a receptor, and when the action potential comes down and hits this receptor, it opens the receptor up and calcium floods out. So that 10 to the minus 7th low level of calcium floating around in the sarcoplasm, all of a sudden that concentration starts to increase. Any calcium greater than 10 to the minus 7th, any calcium coming out of that sarcoplasm, coming out of the sarcoplasmic reticulum, is going to bind to troponin. This is troponin. We've used this slide before in the ultrastructure section. And if you remember, I said that troponin has 3 subunits. This is I, C, and T. The C subunits where calcium binds. Calcium is a fairly large ion. It has a double charge. So think about one calcium has 2 charges and one sodium only has 1. So it's a pretty powerful ion. It floats out from the sarcoplasmic reticulum into the sarcoplasm, and it's anything over 10 to the minus 7th is going to bind to troponin C. Troponin C then goes through a conformational change. It's kind of like the Hunchback of Notre Dame. If I'm standing up straight, I'm going to see if I can walk into the picture here. If I'm standing up straight, then here comes calcium, and it's really heavy, and it comes on the back, my back. I'm going to hunch over, and when I do that, there's a conformational change. In this case, troponin has a conformational change, and here sits tropomyosin. And remember, tropomyosin, and this is a cross section down here, showing each G actin. This is a cross section of it, and this is a cross section of a troponin. And see how it's sitting in the myosin binding site. Well, when troponin makes the conformational change, it takes up more space, and it pushes, see, with calcium, it pushes tropomyosin from the actin-myosin binding site. And in this slide, it has the little starburst there. That's to indicate that, oh wow, it opens that up and it wants to bind the myosin heads. So the important thing is the action potential is the one that results in calcium being released from the terminal cisternae of the sarcoplasmic reticulum. And that increase in calcium from the sarcoplasmic reticulum into the sarcoplasm causes the excess calcium that's been released to bind to troponin, which then allows tropomyosin to shift from the actin-myosin binding site. This slide is trying to show you this concept as well. You see that we have the myosin, we have the thick filament with the myosin heads. The little green means it has an ATP there. We have the thin filament with the purple, in this case lavender, G-actin. Troponin, in this slide, is the ellipsoidal yellow dots, and tropomyosin might be hard to see in this format, but on your slides you should see it as just a thin black string that's going over the actin-myosin binding sites. And so this is at rest. There's no interaction between the thin and thick filaments. Here comes calcium, and calcium is shown in dark blue, and it binds to troponin, which then causes tropomyosin to move from the actin-myosin binding site, and then the myosin heads can come up and bind to the actin-myosin binding site as long as they have ATP bound to them. They then hydrolyze the ATP because they can go up and bind to the actin-myosin binding sites, but unless they have ATP, they can't break. They need an ATPase, which is located here in that myosin head, that then hydrolyzes and breaks down the ATP to ADP, and that's a high energy bond, gives it the energy to contract. When it contracts, it pulls the thin filaments over the thick filaments, so they go towards the M line. And so this is a sarcomere, remember, between two Z lines at rest. And here's a thick and thin filament. And during contraction, when we see the myosin heads flick, so they're going to go like this, they're up, and they go flick, flick. Interesting, I'm sure, for you guys to watch this in the classroom. But we see that pulls these thin filaments this way and these thin filaments this way. And so the sarcomere length gets smaller. I mentioned to you that resting sarcomere length is about 2.5 microns. We know the thick filament, or the A band, is 1.5 microns. And so this I band and this I band, part of the I band, is about 0.5 microns. So we see some movement, and depending on the strength of the contraction, we'll see difference in sarcomere length. One thing that's also interesting, as we move from this relaxed state to contracted state, notice that the A band, where the thick filament is, never changes. The H zone, right here, where there's not overlap, gets smaller, and the I band gets smaller. That's indicating that we're filling in with thick filaments, or overlap of thick and thin filaments in the case of the H zone right there. So we call this the sliding filament theory. So when we talk about contraction, and what I would do if I were you is I would read the textbook, read the all of the read all the text and look at their explanation. This is a figure on page 71 that gives you really the best study guide as far as contraction. So we're going to start at a resting state. We have a nerve action potential arriving at a motor end plate. Make sure that you understand how that action potential moves with that sodium and potassium pump that we talked about on Friday. When the action potential comes down to the motor end plate, there's a point not mentioned here. We're going to add that right now. Calcium, the calcium channel and the axon ending is opened up. Calcium concentration is higher on the exterior of the axon ending. When the channel opens up, calcium floods in. That calcium flux causes acetylcholine to be released from the synaptic vesicles into the synaptic cleft. Then acetylcholine binds to the receptor side on the sarcolemma, which is a sodium channel, and sodium fluxes into the fiber and the membranes are depolarized. Or in other words, the action potential is transferred. The action potential is then transmitted down the sarcolemma, and when it reaches the T-tubules, it goes down into the muscle fiber, and then those T-tubules are right there by the sarcoplasmic reticulum. And the action potential is transferred just the way it was for the axon. It's going to be transferred to the sarcoplasmic reticulum, and that action potential causes the release of calcium. Remember, higher concentration of calcium in the terminal cisternae. Calcium's released into the sarcoplasm, and then the excess calcium above 10 to the minus 7 is bound to troponin. At the same time that that's happening, we have to have an ATP attached to the myosin head, and we have to, then that ATPase is activated, so ATP is hydrolyzed, giving that myosin head energy. Tropomyosin shifts from the actin binding site. These energized myosin heads with the ATP can then reach up, and then they can they can move or hinge and move the Z lines towards the center M line. This action of contraction is a continual formation and breaking of the cross bridges. So it's also important that we talk about relaxation. And we haven't talked about all the steps of relaxation, but I wanted to wait until this slide because we've already talked about the first steps, but I want to talk about how we get those myosin heads released from actin so that they can go back to resting state. To do that, the first thing we have to do is stop the action potential. The action potential does a couple of things. The most important thing that it does is it closes the calcium channel on the sarcoplasmic reticulum and stops the calcium flooding into the sarcoplasm. So cholinesterase is released and breaks acetylcholine down so that it comes off the sodium channel, and then the sodium channel is closed, and the sodium concentration is then with an ATP pump, just like in the axon ending, reestablishes sodium content to be higher on the outside, lower on the inside. When that happens, the action potential is stopped, and the sarcolemma and the T-tubules go back to rest or repolarized. That means if the T-tubules or the action potential has been stopped, well, the sarcoplasmic reticulum also, the action potential is stopped. So the calcium channel gets plugged back up, but we also have a calcium pump that gets activated. Now that calcium pump, as soon as we start leaking calcium, the calcium pump starts pulling calcium back in, because that calcium pump is like that pump, that sodium pump, that was like the bouncer that we talked about on Friday. Same type of activity where that calcium pump pulls calcium out of the sarcoplasm. It's trying to get that concentration of calcium in the sarcoplasm back to 10 to the minus 7. That's its job. Keep that balance, bring calcium back out. And so release of calcium stimulates the pump, and luckily we close the calcium channel. The pump then returns calcium back to the terminal cisternae. So we have calcium then comes off a troponin. As we decrease the concentration of calcium in the sarcoplasm as being taken back up, calcium comes off a troponin. Tropomyosin can then move over the binding site again. Oh, we have a problem though. Tropomyosin can only move across the binding site if we first terminate this actin-myosin cross-bridge formation. The only way to terminate that formation, the myosin heads, think about they're up there, they're stuck, they're holding on. The only way to get them to drop back off and relax is we have to have an ATP come in and bind to the ATP binding site in the myosin heads. Then they'll drop back down and they'll be ready for the next contraction. So if we can get the myosin heads to release, which we need ATP to do, then tropomyosin can return to the actin binding site. But to get everything to slide back, we need ATP, excess ATP, to bind with magnesium. And that's just a stopgap for the muscle to make sure it has a little bit of reserve ATP. It's kind of like having your gas can in your car so that you can, if you run out of gas, you have some fuel. And magnesium ATP complex, always think about it like oil in an engine. It allows the oil or the passive sliding of the filaments back to resting state. And so everything returns to resting state. So I want you to you'll have this lecture on eCampus so you can go back through it and make sure that you understand what's going on. Oh, what's going on here? And so, in summary, what we've done with this lecture and what you need to know is how an action potential moves down a neuron, the steps of contraction, the steps of relaxation. So, if you have any questions, the TAs are here in class. There's a little bit of time for you then to ask some questions. I'm going to ask them to play the CD-ROM to go through this process for the remainder of the class. Because I only recorded 30 minutes of lecture, so hopefully you have 20 minutes left. And I think the CD-ROM will really help you to anchor your thoughts. And after you watch the CD-ROM, we will have it available up in the sensory lab at the computer. And I think that if you can rewatch this lecture after you've watched the CD-ROM, there'll be a lot of things that will anchor in your mind and make sense. So thank you very much, and I'll see you on Wednesday morning on the video recording. Goodbye.